Hello, everybody. I'm Didier BOICHARD, Senior Scientist at INRAE, and I'm very pleased and honored to chair this seminar, this webinar on epigenetics and methylation marks. And I thank EAAP and Illumina for this opportunity. We will have 4 talks, but I will come back on that a little bit later. Just a little bit of practical things. You will not be allowed to ask directly your question in a loud way. So you are invited to ask your question in the chat, and I will read them and try to answer with the rest of the panel. And we will have a question and answer after each talk, and also at the end of the webinar. So a few words about to introduce the subjects. So in 2007, the bovine SNP chip was launched, and followed by many other chip versions, generic or custom, with various densities and in a number of species. And you all know how these tools have revolutionized the selection, which has become genomic. And now we are 18 years later, and a new tool has appeared for the first time for farm animal species, the bovine EpiChip, the RUMIGEN EpiChip. And this tool is able to estimate the methylation rate for a large number of CpG sites in the tissue sample, so we can anticipate it will be used on a very large scale, depending on the possible applications. So to present the topic in this webinar, we will have three main talks. Hélène KIEFER will present the EpiChip and its potential for prediction. Corentin FOUERE from Alliance will show you the interplay between genetics and methylation in bull sperm. Marie-Pierre SANCHEZ from INRAE will show you the first result of the EpiChip for phenotype prediction in dairy cow. And I will conclude on a vision of the potential future of animal breeding integrating this information. So I invite you, Hélène, for the first talk. Perfect.
Okay, good. Sorry for the delay. So thank you very much for the introduction, and thank you for the opportunity to present our work, which has been done in the framework of the RUMIGEN project, which is a European project. And my presentation is entitled The Potential of Sperm and Blood Epigenome in Selection and Precision Farming in Cattle. So because this webinar is co-organized with Illumina, I have to show you this screen. So a few words about epigenetics to introduce the topic. So epigenetics alters gene regulation without modifying the DNA sequence. So there are 3 main epigenetic mechanism: DNA methylation, which covalently modified genomic DNA, histone modifications, and non-coding RNAs. And in promoters of genes, the absence of DNA methylation together with activating histone marks are usually associated with open chromatin and with active transcriptions, while the presence of DNA methylation together with repressive histone marks are associated with compact chromatin and transcription repression. So at the cell level, these epigenetic mechanisms represent the memory of cellular identity, and they are transmitted through mitosis. And at the individual level, they represent the memory of environmental exposures and can also mediate long-term effects on the phenotype. And lastly, there's the possibility that these mechanisms are transmitted to the next generation. Among all these mechanisms, we focus on this webinar on DNA methylation, which targets CpG denucleotides in the genome. DNA methylation is stable, it is easy to quantify at different scales, and importantly, it can be analyzed as a sequence polymorphism after chemical or enzymatic treatment, which converts unmethylated cytosines into thymines, while methylated cytosines remain as cytosines. So from this information, it is easy to calculate a methylation percentage. So depending on the question, it's possible to analyze DNA methylation at different scales. If we are interested in one or few CpGs, we can use techniques such as pyrosequencing. And there are also DNA methylation arrays that have been designed for humans, for mouse or other mammalian species. And these methylation arrays target a specific panel of chosen CpGs. And finally, it's possible to investigate all the CpGs present in a genome using sequencing-based approach, such as whole genome bisulfite sequencing or Oxford nanopore technology sequencing, or to target the analysis on a CpG-rich region. And this is the case of RRBS, reduced representation bisulfite sequencing. And in my presentation, I will introduce the RUMIGEN EpiChip, which is the first cattle-dedicated DNA methylation array. It's a median density array which targets 44,000 CpGs. And you will also heard about RRBS in the presentation and in the following presentation of these webinars. So the RUMIGEN EpiChip is a Illumina DNA methylation bit chip, which has been designed to achieve the scientific goals of RUMIGEN, but to be also efficient for routine use and to be cost effective, and to be useful for the international scientific community and for future projects. So we used, to design this EpiChip, we used a two-step approach. The first step was a data accumulation step, where we accumulated data produced using RRBS, nanopore, and also publicly available WGBS data, mainly focused on sperm, blood, and the embryo, because it was the main targets of the RUMIGEN projects. And then we invited contributions on other traits and other tissues. And finally, we added generic positions in order to cover most genes of the cattle genome. And next, this phase was followed by a data selection step, where we used data from the 1,000 bull genome projects in order to filter out all the CpGs or probes, some probes with overlapping polymorphism. We also took advantage of our large RRBS data collection in order to select the most relevant CpGs among nearby CpGs. And we took advantage of functional annotation data available through the FarmGTex and the FANG initiative in order to select a relevant panel of 44,000 CpGs. This is the final content of the RUMIGEN EpiChip. So half of this content has been provided by RUMIGEN. 25% has been contributed by external contributors, and 25% target genes and functional elements. And in total, 23,000 unique gene IDs are targeted by the EpiChip. The EpiChip mainly targets CpGs that are located within genes or in regulatory regions such as transcriptional start sites and promoters. And not so much CPGs are located into overlapping repeats or in CPG islands in gray here, because these CPG islands usually show poor variability across individuals. We also investigated more in detail functional regulatory elements, and it turned out that a large number of CPGs on the RUMIGEN EpiChip was associated with chromatin states related to transcriptional regulation in blue, and using data provided by the BOVREG European project, it was shown that more than 9,000 CPGs represented promoters, either cell-specific promoters or pervasive promoters, and more than 4,000 CPGs were located into enhancers. And interestingly, a small share of these enhancers show evolution, show a large degree of conservation across evolution. So next, we had a tool showing a very relevant content. And next step was to validate, to appreciate the technical validity of this tool using a validation trial. And the first objective was to investigate the repeatability of the tool. For that purpose, we used 4 different samples, 2 blood samples and 2 semen samples, and an important number of replicates of these samples. And all the epigenotyping steps have been performed by JD Biotech facility in France. So here are the results. Each little red square shows the correlation coefficient between 2 pairs of replicates. And overall, the average correlation is above 0.99 across technical replicates, demonstrating that the tool has a high level of repeatability. We next investigated the correlation with RRBS as a gold standard using the same subset of samples. And here are the results. So the x-axis show the DNA methylation percentage measured using RRBS, and the y-axis the DNA methylation measured by EpiChip, and on blood and on semen. And across all these technical replicates, the correlation coefficient was above 0.90 between EpiChip and RRBS on the CPGs covered by both technologies, which show very good correlation with existing technologies. So the technology show good repeatability, a good correlation with existing and independent technology. So the next step was to investigate its ability to target a known DNA methylation value on an accurate way. For that purpose, we mixed samples of blood of semen with different increments to create artificial mixes. And we measured DNA methylation values with the EpiChip on these different mixes. And in parallel, we also calculated the expected methylation values in all these different mixes from the DNA methylation values measured from the pure cell types. And there are more than 1 million points represented on these graphs, showing the correlation between the expected methylation value and measured methylation value. The coefficient, the correlation coefficient was above 0.98, and the median deviation between expected methylation value and measured methylation value was only 2%, showing a very good accuracy of the tool. Next objective was to evaluate the interoperability of the tool between batches and between labs. And for that purpose, the samples were analyzed in 2 different batches by 2 different labs. So GD Biotech, as shown previously, but also Xenetica Fontao in Spain. And the results are shown on this principal component analysis. So the first dimension highlights the high difference between blood and semen samples. The second dimension highlights technical variability, which was mainly driven by the difference between the two labs. And the third and fourth dimension highlights the difference between the 2 samples for each cell type. So these results demonstrate that the variability is due to both biological factors and to technical factors. And this has been also widely described for other DNA methylation bit chips. Next, we focused on imprinted regions. So the methylation status at these regions control the monoallelic expression of genes. And in somatic cells, it is expected to have 1 methylated allele and 1 unmethylated allele. And as expected, we measure a methylation rate of 0.5 for all the investigated regions. And in contrast, using sperm, we measured very low methylation level for all the paternally expressed imprinted genes, and in contrast, a very high methylation level for the only maternally expressed genes that we investigated. So these data obtained on imprinted regions was in good correlation with the results that were expected. And finally, we investigate the ability of the tool to highlight cell type specific patterns, and especially targeting blood. So we purified the main cell types that are included in whole blood. And using a very specific subset of CPGs, we were able to nicely separate these different cell types. So this raises the possibility to infer blood formula from these specific CPGs in order to correct the DNA methylation values obtained on whole blood from this blood formula. So in conclusion for this validation trial, we obtain DNA methylation measurement which were highly repeatable using the RUMIGEN EpiChip. And the measures were well correlated with sequencing-based methods. And the tools also demonstrated a very good ability to target a given methylation value. There was technical variability due to lab and mainly lab, but also batch effects, as it was also reported for other methylation arrays. But most importantly, we were able to detect biological variability due to cell type specificity or also to interindividual differences using the RUMIGEN EpiChip. So first, I would like to thank all the contributors to the design and to this validation assay, and most especially Valentin Coste. And next question is, what are we going to do with this new tool in RUMIGEN? And I will try in the following slides to give you an overview about this question. So first, many steps in a bull's life can impact the sperm epigenome. Indeed, the bull can be issued from embryo technologies or from high-producing dams under strong metabolic demand. Then the bull is recruited by a semen production center, and the environment during the growth phase of the bull is important. And then there's the impact of early semen collection as soon as puberty has been reached, the impact of semen processing treatments such as sexing, dilution, packaging, etc. And it is important to understand how these different steps can alter the sperm epigenome and to what extent these alterations can be transmitted to the progeny, because due to the widespread use of artificial insemination, semen is widely spread goes to many herds. And in parallel, many steps in a cow's life can also impact the blood epigenome. This graph represents the DNA methylation level during development. And this DNA methylation is highly variable during the very early steps of in utero life. And then it tends to stabilize in somatic cells. And in parallel with this stabilization, there's a decrease of genome plasticity. This means that the impact of environmental exposure is higher when they occur during the very early steps of the life. And having said that, it means that if we collect blood sample on an adult producing cow, then we may have the ability to integrate all the effects that about gamete qualities, embryo technologies, dams metabolism and environment, disease, nutrition, and so on, that have impacted the animal during his history. So the questions we ask in RUMIGEN is related to epigenetics and how it can improve genetic evaluation and individual prediction. So today, phenotypes are mainly predicted from breeding values, and there's a big black box that contains all the effects of the environment. And we investigate if transmitted epigenetic variations exist in cattle, and if sperm epigenome explain part of the heritable fraction of complex traits. Because if we can measure transmitted epigenome in the sperm of the father, then it will allow to refine a prediction model. Another question is whether epigenetics can reflect the environment black box to improve individual performance prediction. And if so, the epigenome which has been acquired by the cow during her life history and measured on the blood can also help to refine phenotypic prediction. So to address all these questions, we use a very unique experimental design in which we have 500 marketed Holstein bulls with different ejaculates per bull, and 5,000 Holstein cows with genotypes and phenotypes available. And most of these cows are daughters of the bulls we have. And I would like to thank very much all the breeding companies and also INRAE who contributed to obtain this large experimental design. And so using the epigenotypes of bulls, which has been acquired by RRBS on sperm, we want to address the plasticity of sperm epigenome, the genetic control of sperm epigenome, and how the sperm epigenome can impact bull fertility and the phenotype of progeny. And using the epigenome of cows, which has been obtained on blood using the EpiChip, we want to investigate the impact of cow epigenome on their own performances. And the next talks during the webinar will address the genetic control of sperm epigenome and the impact of co-epigenome on their performance. And then I will leave the floor to Corentin and Marie-Pierre, which will illustrate their result using a specific subset of this experimental design. And with that, I would like to thank you for your attention, and I'm ready for questions. Thank you very much.
Thank you, Hélène, for this presentation of the chip and its potential use in RUMIGEN. Not potential, its use in RUMIGEN. We have time for a few questions. So a question of André again. What would you say should or could be the best tissue to target for routine testing in order to benefit from this new tool for methylation profiling? Sperm for bulls and blood for cows? Or is there any other tissue to be considered in the future?
So Thank you for the question. Probably easily accessible tissue would be best for routine analysis. For that, sperm and blood are quite good, but we can imagine using, for instance, some hair follicles or saliva from epithelial cells collected in the saliva, or milk, or maybe ear biopsy used at the time of genotyping. And there are several things to consider. One thing is the homogeneity of the sample. And the other thing is what is the good time to collect this sample? Because, for instance, sperm can only be collected from sexually mature bulls, and milk also for producing cows. So these are different things to consider.
Yes, because you have to remember that the methylation marks vary according to tissues. So you have to be sure that if you measure in one tissue, you can correlate that to methylation results in another tissue, or at least with the performance you want to predict, whatever the origin of the tissue. So quite a lot of work to build this this system to translate from one tissue to the next. Maybe, I don't know if it's possible, but it would be. We are here to dream a little bit also, and to work a lot. Another question from Laurent Boulanger. Hi, could it possible in the near future to create a robot to diminish the internal laboratory variability results in the use of the chip?
So I cannot answer this question, but regarding the interlaboratory variability, it is quite well documented, and there are tools, bioinformatics tools and statistical tools to correct from this variability. This will not replace a good experimental design. So if you want to decrease this variability, it's best to analyze all the samples in the same lab if possible, and on the same slide to mix different comparisons we want to address. It does not reply directly to the question, Buff. But
I know that methylation is more difficult because it is a continuous variable, but at least for SNP, the interlaboratory variability is not a big problem. I know that it is easier, but anyway. Another question. How did you analyze control on this array with Genome Studio, I guess?
No, it's not possible to use Genome Studio with this array. So we analyze control directly using the raw fluorescence data.
Yes, but I guess in the labs they use Genome Studio to prepare, to translate the signal into.
They directly. What we get is IDAT files from the lab.
It sounds like you used Sesam for analysis.
Yes, exactly.
Okay. Any other question? If not, given that different perturbation, nutrients, heat stress, cold stress may cause different specific methylation, does the 44K chip captures methylation that result from many different types of environmental perturbations? And is there a form of linkage disequilibrium that helps capture methylation which are not on the chip?
So this is a very good question. And of course, we do not pretend to capture all the methylation variability of the genome within this single chip. And this is why we also use complementary approaches based on sequence to cover most of the genome. The chip is very good to address questions which require high important statistical power and large population studies. For instance, to perform epigenome-wide association studies or analysis such as Marie-Pierre will present later. But to capture all the variations of the genome, it's probably not the best tool, and we will need anyway more and more sequencing data in order to investigate its variability and to design an improved version of this chip. And for the second part of the question, I think Didier has planned to speak about it in the conclusion, so I will let this question to Didier.
Yes, but it will be very short. But yes, there is a form of linkage disequilibrium, partly related to the physical distance, partly related to functional reasons. You have several methylation marks which are grouped in order to regulate the expression of the genes, and this generates a form of association. But we have to discover that in more details. Another question, how many CPGs, including EpiChips, are located within the known SNP?
Theoretically, no. Theoretically, because we filtered out CPGs with overlapping SNPs using data from the 1,000 bull genome. It's possible that unknown SNPs still remain.
Yes, but they are rare, I would say. Very rare in terms of number and very rare in terms of frequencies because they have been eliminated. And that's a very critical question because if there is a SNP in the CPG site, of course, the derived allele cannot be methylated. And if the original allele is 100% methylated, therefore you observe Mendelian transmission with 0, 50%, 100% methylation. And that's not the information we want to see because it's already tested by the SNP. So they were voluntarily eliminated.
Okay, thank you, Didier. Hello, everyone. Do you see my screen? Yes.
Yes, that's okay.
Okay. So first, thanks for the invitation to present this work. So I'm a third year PhD student between Alliance and INRAE. And within the RUMIGEN project, I worked on the genetic regulation of sperm DNA methylation in cattle as introduced by Hélène. So here is the disclosure statement. And then I will be fast on this slide, but it's just a reminder of what is DNA methylation. So it's the most studied epigenetic mark. It's not the only one, but it's the most studied. And in mammals, it's the methylation of the fifth carbon of the cytosines, so this, and it only or mostly occurs in cytosines phosphate guanine groups, so the so-called CPGs. And it's of particular interest as DNA methylation provides a link between the DNA sequence, the DNA information, and the phenotype. It can influence the gene expression. And also it can be, and it has been shown to be influenced by environmental factors, such as heat waves, for example. But here, the aim of this work was to understand the genetic architecture of DNA methylation in cattle. And what DNA methylation information we used. So we used one specific tissue, which is semen, And compared to somatic tissues, it's a tissue that is mostly highly methylated. We had DNA methylation information for a bit more than 400 Holstein bulls. And the technology used was not the EpiChip, it was the reduced representation bisulfite sequencing. And briefly, it gives methylation rate from a number of read or unread CPGs, cytosines, for a given CPG site. As Hélène previously mentioned, it has a biased genome representation. So first, the CPG dense regions are more covered than the rest of the genome. And also we observe that the end and the beginning of chromosomes are much more covered than chromosome centers. And from this RBS, we had information for about 2 million CPG sites, which represent approximately 7% of all the CPGs in the bovine genome. Well, not all those CPGs are of interest for our study, because in sperm, but also in somatic tissues, most CPGs are either fully methylated or unmethylated between bulls. So they are represented on this graph. So this graph shows the methylation mean across the 400 bulls and the methylation standard deviation. And on the top and on the right side, there is the distribution of the CPGs. And as you can see, there is a large proportion of CPGs that do not demonstrate any variations. But we did not select them. We only select CPGs that are above the red line. And the red line is at standard deviation of 5%. And so we select those CPGs, and it represented about 167,000 CPGs, so about 8% of the 2 million CPGs for which we had RBS DNA methylation information. So on the other side, we were interested in the DNA information. So all the bulls used in the study were genotyped using a medium density chip, so the EuroGMD. Then we impute up to the whole genome sequence level using R-square of minimac of at least 0.3. And we also selected only variants that had MAF above 3% to ensure that we had only at least 20 copies of the rarest allele. So this gave us around 13 million variants, mostly SNPs. And on the other side, as I was saying, we had DNA methylation. The important step, but it was already discussed in the questions, is to remove SNPs, CPGs that colocalize with SNPs, as the bisulfate conversion will put noise into the DNA methylation information. Also, we kept only only CPGs that had at least 80% of information across bulls, and we corrected the DNA methylation information of each CPG for the age at collection of the bulls. So we had those 167,000 CPG sites. Then we conducted genetic analysis. So we did GWAS, and it was only conducted on autosomes. And from those GWAS, we estimate heritabilities, and we also mapped MEQTL. So MEQTL means methylation QTLs. The software we used was GCTA, and we did one GWAS for each CPG, for each of the 167,000 CPGs, with the DNA methylation being a function of a polygenic effect, the SNP additive effect plus a residual effect. And when we mapped the methylation QTL as all the molecular QTLs, they have a location on the genome, and we classified the methylation QTL depending on the location from the CPG to the QTL. When the CPG was localized, when the QTL was localized within 1 megabase of the CPG, we classify them as cis MEQTL. And when the distance was above 2 megabase on the same chromosome or on another chromosome, we classify them as trans MEQTLs. Here we move into the results, and this graph shows the distribution of all heritability estimates for the 167,000 CPG sites. So the estimates are based on the genomic relationship matrix. And as you can see on this graph, a lot of CPGs have null heritability, but some have pretty high heritability. So it goes from 0 to 1, and the average heritability is 0.26 across all CPGs. Staying on heritability, we also wanted to look whether there are several genomic regions that shows enrichment or depletion for highly heritable CPGs. But first, I will define shortly the CPG landscape annotation. So in the genome, the CPGs are not equally distributed. There are regions that are called CPG islands that are rich in CPGs, and around those regions, we usually define a 2 kb region, which is called the CPG shore, then a 2 kb region, the CPG shelves, and between those regions, we call the genomic region the open sea. And from the CPG island up to the open sea, the richness in CPG site decreases. And on this graph, so this graph shows the enrichment of CPG with high heritability compared to all the CPG analyzed. And you can see that the open sea, so the regions that are poor in CPG density, and the intergenic regions are enriched for heritable CPGs. We call heritable CPG when the heritability was above 0.2. And for the regions that are, so the transcription start sites 200 and transcription start site 1500 that are representing the promoters, there are regions where we observe depletion for heritable CPG. It has been observed also in a human study, but we do not have any clear biological explanation for that. Moving from heritability estimates to methylation QTL mapping. So this first graph shows the distribution of the QTLs we observed from the mapping. And first, we had about 1/3 of the CPG we analyzed for which we found cis MEQTL. We had 3% of the CPGs that showed trans MEQTL, and we had 1% that had both cis and the trans MEQTL. And when we only consider the cis MEQTL, as they are on the same chromosome, we can plot the distance between the CPG and the cis MEQTL, and specifically the tops, the most significant SNP. And as you can see on this graph, the distance between the top SNP and the cis and the CPG is often very close. For 1/3 of the CPG, it's below 33 kb. From all the CPG we analyzed and the MEQTL we obtained, we wanted to look at the distribution of those QTLs compared to all the SNPs analyzed. And I will start this graph with the first line. And so this represents the CGI, the CPG island. And in green, it's the cis MEQTL top SNPs on all the SNPs analyzed. And in orange is the trans MEQTL top SNP on all the SNPs analyzed. And it represents the odds ratio. So for the CPG islands, we observe that the SNPs that are associated with an MEQTL, whether it's a cis or a trans, they are overrepresented compared to all the SNPs analyzed. We also observed enrichment of MEQTL for transcription start sites, the TSS regions, 5' UTR, 3' UTR, and also attack-seek peaks. So attack-seek peaks represent the regions of open chromatin, which often colocalized with regulatory elements, regulatory regions. So it's kind of expected to have enrichment in those regions that are active in the genome for QTLs that, for example, regulates methylation. And overall, we observe a shared pattern between cis and trans top SNPs, even if the trans were often more pronounced than the cis. For the trans MEQTL SNPs, we found that several trans MEQTLs were associated with multiple trans CPGs. And we defined these regions as trans MEQTL hotspots, and they are represented by the peaks on the graph. So this graph looks like a Manhattan plot GWAS, but it's not. It represents the number of times each SNP is associated with a CPG. And for example, if we take chromosome 2, there is a peak. And this dot here represents SNP that is associated with more than 500 CPGs that are localized at distance. So the trans CPGs that are localized either on the same chromosome but at a long distance or on another chromosome. And in fact, if we look at whether where those CPGs are, they are on many different chromosomes. From those peaks, we also try to look for candidate genes in those regions, and the red line here defines SNPs that are associated with at least 30 CPGs, trans CPGs. And here I put some candidate genes, and so for several and almost all trans SNP hotspots, we found that the genomic location overlap with genes involved in epigenetic regulation. So whether they were histone modifiers, for example, KATE2B or HJK1, and other were transcription factors. And some are known to interact with DNMT3. So it's a DNA methyltransferase that makes the de novo methylation. For example, SPOC D1 or TCL1B. So to sum up, we showed this work shown that genetic variation influence sperm DNA methylation. Most of the QTL we found were cis, so they were located close to the CPGs they affect. We also found for the trans MEQTL some regions that target multiple CPGs with, for most of these regions, candidate genes that include histone modifiers and chromatin remodelers. So we might ask, we might wonder if DNA methylation might be a result of change in the chromatin conformation. And overall, the results we observed are in line with the existing literature, even if there are not much methylation QTL mapping in livestock species. It's mostly done in humans and mostly on blood tissue. And as Hélène said, and Marie-Pierre will present, DNA information is envisaged to be used to better predict phenotypic traits or even breeding values. So there is a need to unravel the genetic determinism of DNA methylation. And finally, we hope this study contributes to improve our understanding of the genetic architecture of sperm methylation. With that, I would like to thank you for your attention and thank the RUMIGEN project and all the partners. And this work, if you would like more details, it has been recently published in BMC Genomics. Thanks.
Thank you, Corentin, for this very comprehensive talk. So we already have two questions. The first one from Alina and G. Paris is quite general. I will shift it to the end of the webinar, and then we will come back on it, on the conservation of methylation marks between population and species, especially. The second one from Stanislas Kaminski. DNA originate from more than 400 bulls, where these bulls in similar age, their semen were collected in similar frequency, semen production traits were similar in level, so semen volume, motility, were they held in similar environmental condition, feeding, well-being condition, etc. So, Corentin, can you give more information about the comparability of the methylation data in sperm?
Yes, so there were some age difference. There were some very old bulls and some very young bulls.
In majority, they were young.
Sorry?
In majority, they were young.
Yeah.
Yeah.
Well, the semen comes from commercialized straw, so there might be difference in the semen parameters, but at least they respect the global criteria. So they are not very deviant for semen parameters, and they match the known criteria for spreading. And for the breeding conditions, we do not have a lot of information, but the bulls come from maximum 3 locations, and usually they are protected against like strong environmental effects. So, well, it's not a perfect design, but I think it's quite homogeneous conditions.
And you adjusted for some factors.
We adjusted for age at collection.
Age at collection. Okay.
And using classes and not a linear regression.
It's not done by you, but in previous studies, the team of Hélène studied quite extensively the relationship between methylation and fertility, male fertility, and semen characteristics.
Yes.
So you can find that in different publications. A question from Husinu. Can you explain why did you exclude the intrachromosomal CpG SNPs that also had a cis-MeQTL from further trans-MeQTL analysis? Do you understand the question?
Yes. Well, when the trans-hemicuteal was close to the CPG location, we had many of those, and for I think more than 99% of the time, there were also cis-hemicuteals. So well, the trans-hemicuteals that were localized close to the CPGs often were false trans-hemicuteals, but they were cis-hemicuteals. But due to the LD, they were still observed at some distance, and well, we exclude the trans-hemicuteal when there was a cis-hemicuteal. So I'm not sure I'm answering the question right, but well, in cattle there is a quite high LD and sometimes up to several megabase. So often when we observe a trans-hemicuteal at a close distance, it's false cis-hemicuteal.
But it's simply due to the fact that you have a top SNP, which is a real cis-QTL.
Yeah.
And you have other SNPs less significant, but it's a little bit far away, and those are excluded because the signal is probably due to LD.
Yes.
Okay. Okay, a few minutes still available for question. Otherwise, I can come back on the more general question. Alina G. Paris. Within sample types, blood, for example, are you or do you expect to see significant methylation differences across bovine species? So species or breeds to be clarified. For example, would the DNA methylation in blood of a Holstein cow be significantly different from a different species? This question is open to anyone who would like to address it.
I can reply on that because
Okay.
we made some comparisons between breeds, cattle breeds, mainly in semen, but also in blood. And indeed, we see very important differences between breeds. And this may be explained by what Corentin has shown during his presentation. There are some methylation marks that are controlled by genetics. So any difference between breeds can underlie also DNA methylation between breeds. And even after having filtered out putative SNPs between breeds or all the SNPs, we still see very important differences between breeds in two cell types, so blood and semen.
And maybe a more general comment. There are several types of methylation marks, I would say. Those which are extremely regulated with no methylation or 100% methylation. And those marks are the most numerous. And there is nearly no individual variability on that. And for, let's say, I don't know, maybe 10% of the methylation marks, you have some variability. And usually we focus on this variability between individuals. But if we compare the 90% of the marks which are fixed, I guess they are still fixed across population. Don't you think so?
Across breeds, you mean?
Across breeds, maybe even across species. You have some patterns which are conserved across species.
Probably because pan-mammalian methylation array has been designed based on this conservation. So it's, yes, you're probably right. I would not say anything about the percentage because I don't know, but
no, for me, the percentage is just what we observed in RBS. the proportion of nearly fixed methylation marks is quite large. And they are not the same from one tissue to another. In fact, they are responsible for the differentiation. That's the basic role of the methylation.
Thank you, Didier. Good afternoon, everyone. I'm going to present a study on the use of the RUMIGEN EpiChip data to predict dairy cow performance. So disclosure statements first. As Hélène just mentioned, the DNA methylation EpiChip was developed in the European RUMIGEN project, and it is a powerful tool to explore how epigenetic factors influence dairy cow performance. And the objectives of the present study are to assess the contribution of epigenetic biomarkers to phenotypic variation in cows by estimating first the percentage of variants explained by methylation, and second, by estimating the impact on the accuracy of estimated breeding values, EBVs, in a validation population. So for that, we had almost 5,000 Holstein cows with EpiChip methylation data. These cows were born between 2013 and 2023, and they were raised in 259 French farms. For part of these cows, we had also different phenotypes related to milk production, further health, fertility, and morphology. Just to have an overlook of the methylation data of the EpiChip, as Corentin just presented before for RRBS. And you can see also the mean variability, the mean methylation rate plotted against the standard deviation. And in the EpiChip data, about 5% of methylation marks show little or no variability between the cows. So first, we applied different filters on the data. First, it doesn't work. First, by retaining marks with less than 20% of missing data, and for the cows, we applied the same filter. And for marks, we retain only the most variable marks, that is to say the mark with a standard deviation higher than 1%. And in the final data set, we had more than 38,000 marks for 4,922 cows. And we corrected the methylation rates for leukocyte composition. And at the end, we had a methylation matrix M that contained the percentage of methylation for each mark Y and each individual J. From this matrix, we calculated an epigenomic similarity matrix S by calculating the coefficients using this formula that was developed by Van Raden for SNPs. In this formula, all the terms were standardized by the mean variability of methylation, meaning that the marks with higher variability were given more weight. And in this similarity matrix, we observed that three cows were outliers, and we removed these cows. So you can see here the number of marks and the number of cows we had in the final data set we used for the analysis. For estimating the accuracy of EBVs, we divided the whole population into a training and into a validation population. In the validation population, we kept the youngest cows So we had more than 3,000 cows in a training population and almost 800 cows in the validation population. When we looked at the sires of these cows, we found that 70 sires had daughters in both training and validation population, and we decided to separate sire families in both population. So you can see here the results we had for milk production, for example, with 3,000 cows in the training population and more than 500 cows in the validation population. And in this table, you can see the number of cows in each population for the different traits we analyzed. Then we applied two different models. Two different models. In the first model, we have the adjusted phenotypes, so phenotypes corrected for non-genetic effects that were explained by the fixed effect of birth year, the random genetic effects, and the random residual effects. And in the second model, we had an additional effect, which was the random methylation effects that were estimated using the similarity matrix presented before. And for each model, we estimated irritability, which was the ratio between the genetic variance and the total variance. And in the second model, we estimated the percentage of variance explained by methylation, ME2, which was the ratio between the methylation variance and the total variance. So you can see here the results we obtained for the percentage of variance explained by genetics and epigenetics. So for each the traits we analyzed, you can see the percentage of variance explained by methylation in dark blue, the percentage of variance explained by genetics in medium blue, and the percentage of variance explained by residual in light blue. So we can see that for 14 traits of the 33 traits we analyzed in total, we had a percentage of variance explained by methylation that varied from 1% to 15%. For example, here for milk fat yield and milk fat content, we had a value of 1%. This value was 3% for somatic cell score and 1 fertility trait here. And for 10 morphology traits, we had values between 1 and 15%, and the highest value was obtained for dairy farm. Here you can see the results we had for the accuracy of EBVs that were calculated by the correlation between the YDs, which are the phenotypes corrected for non-genetic effects, and the EBVs we estimated in model 1 in red and in model 2 in green. And as you can see, for 8 traits, we observed again with the second model for the accuracy of EBVs, and this gain varied from 0.5% for milk fat yield to 6.7% for dairy farm. So to conclude, with the similarity matrix constructed with Van Raden method 1, which gives more weight to most variable marks, we find that the epigenetic effect was higher than 1% for 14 traits and explains up to 15%. We also observed an improvement in the accuracy of EBVs for 8 traits, and this gain varied from 0.5% to 6.7%. However, the EBV in this preliminary study, the EBVs were estimated using pedigree data, as not all cows have been genotyped. And only a subset of the dataset was considered, since performance data are not yet available for all cows with EpiChip data. And in addition, we used phenotypes adjusted for non-genetic effects, which could have an impact on the results. So we plan to conduct the same study by including the complete dataset, that is to say about 5,000 cows with genotypes, epigenotypes, and performance, by estimating the breeding values using the 50K genotypes with a genomic relationship matrix, and by testing different methods, in particular methods with a selection of variables. And also we will perform analysis with the raw phenotypes, that is to say phenotypes non-corrected for non-genetic effects. So that's it. So thank you for your attention. Thank you to all my colleagues who contributed to this study, and to all partners of the RUMIGEN project.
Thank you, Marie-Pierre. These results are quite fresh, I would say. And as you can see, we don't have all the phenotypes for all the cows, so only the phenotypes of 4,000 cows have been analyzed. Est-ce que tu peux couper le partage, s'il te plaît?
Oui.
Merci. So these studies will continue with all the phenotypes, of course, and with different methods. I have a question from sorry, I cannot see the name, but for stature, there was no epigenetic effect, but for other type traits, showed very high. How can you explain that? For you, Marie-Pierre?
Yes. Yes, in fact, we analyzed different morphology traits, and they were not all related to stature. So it is not completely discordant to observe that we have effects on some morphology traits and not on other.
And also to complement, it's only a feeling. It would be worth to verify, but I have the impression that the most affected traits by the traits the most affected by methylations are those which are sensitive to environment. So the feet and legs, and also the body condition scores, the dairy form, and so on, which are in contrast, the stature is highly irritable and under genetic control. Maybe, I'm not sure. Another question from Stanislas Kaminski. Have you identified blood CPG from cows with a single blood sample, or many times from several blood samples collected, for example, in subsequent lactations?
In this dataset, we have only one sample per cow. But
Yes.
the cows, the age of the cows varied.
In fact, we were limited by the number of analyses using 5,000 chips. It was already quite expensive. And of course, it would have been possible to sample several times and mix the samples before the analysis, but even the sampling procedure was quite tedious to obtain. You also notice that the number of herds was quite large, because for part of the design, we